Okay, so good afternoon, everyone, and greetings from Nature Kenya. My name is Richard Kipngeno, the Birding and Membership Officer, and I will be the one moderating our talk this afternoon. It is our first talk for the year 2022, and we really thought it wise that we venture more deep into insects and get to learn more about mosquitoes. So just to let you know, we are recording this presentation, and then later on we'll also be able to share in our YouTube channel. You can always revisit. And also today we are live on Facebook in Nature Kenya page. And I will request that we all remain muted as the presentation goes on. And then if you have any concern, any question, any compliment that you want to raise, you can always do that through the chat box. We will strive our level best to ensure that we go through each and every at the end of the presentation. So off we go. And today our speaker is Mr. Laban Njoroge, who is a conservationist and medical entomologist with a big passion for forensic entomology. He is currently the keeper or curator of invertebrates at the National Museums of Kenya in Nairobi. In entomology for conservation, his key interest is in aquatic invertebrates, particularly mayflies, stoneflies, caddisflies, and dragonflies. However, his formal training was in the area of medical entomology, and which he continues to pursue alongside the other interests. His interest in forensic entomology is seeing him develop a training curriculum for the investigative departments in Kenya together with the DCI, the Directorate of Criminal Investigation. He will discuss with us about mosquitoes generally and tell us why and how he rears them. Thank you very much. The floor is yours, Mr. Laban Njoroge. Karibu sana.
Thank you very much, and I appreciate that introduction. I'm not new to Nature Kenya. We've interacted quite a lot, and we've talked about different topics. So I'm sure most people here have met me before. So it's good to meet virtually. You actually feel closer to me than if we met in a room. I'm almost tempted to reach for my barakoa or the mask because of how close you seem to be to me. So we're gonna interact in this talk, and I'll tell you the outline of the talk. We generally discuss about the mosquitoes, and then we'll touch a little bit on the taxonomy or the classification of the mosquitoes. And then, like Richard mentioned, we will see why some people rear or breed mosquitoes. You know, when you think of mosquitoes, I know most of you, just when you say mosquitoes, all you think of is getting a can of pesticide, a can of doom, or if there's a slipper, you know, pair of slippers nearby, to just mash them on the wall or to use your palms. You know, that's what most people think of mosquitoes. So it's sort of weird when you hear that there's some people who even breed them. There's some people who even rear mosquitoes. So I think there's a good reason why they do this. And in the course of the talk, I'll be trying to explain why some people are weird to that extent. So like Richard said, my formal training is in the area of medical entomology, but I've developed interest in the other areas that he mentioned. So I'll go straight to the talk, and we'll discuss mosquitoes together. Okay, so I think it's a good starting point, posing the question, what are mosquitoes? And you probably are saying, of course, these are insects. Yes, they are insects, but what kind of insects are these? I mean, are they beetles? Are they butterflies? Are mosquitoes grasshoppers? Yeah, what kind of insects are they? Now, these are flies, but I thought all insects are flies. Fine, then these are what we call true flies. Now, true flies are insects that belong to an insect order we call Diptera. And one key characteristic of these flies, we are calling true flies, is the fact that they have only two wings. They have one pair of wings. Yeah, you know, most insects will have two pairs of wings, and therefore 4 wings in total. Yeah, but these insects that we are calling dipterans, or members of that insect order we call dipterans, in which the mosquitoes belong, they've got only 2 wings, or one pair of wings. Now, there are insects that are closely, they closely resemble insects, and they take all the beating. They are on the receiving end when they're spotted by most people. So when you see them on the walls in your houses, all you think is the various things that I talked about, either can of doom, you take a pair of slippers to smash them on the wall. Yeah, but they are harmless. Yeah, they do not bite. They do not probably even notice you. But because they closely resemble mosquitoes, they take the beating. They are on the receiving end. And one of such is this creature here that we commonly see when it rains, and you find it coming so much to light. It's highly attractive to light at night. And it's got very long legs that very easily come off when you touch them or when you try to grab it. The legs very easily fall off. Now, this insect, which people call big mosquito, is indeed a crane fly. If you like, you can call it the daddy long leg. Yeah, simply because of the long legs it has. It is a harmless mosquito, and this is how, a harmless insect. It's not a mosquito, it's a harmless insect, only that it resembles mosquitoes. It's got no proboscis, which is that long biting and sucking mouthpart that mosquitoes possess. So if you keenly look at it, you'd realize very easily that it doesn't have a biting proboscis. And then it's much, much larger than, the mosquitoes. So probably those are some of the differences that can help you identify it very quickly. Now, the other group is what we call the lake flies. And lake flies are the sort of insects you find in parking lots very early in the morning, towards the evening, when the weather is warm. And you find them in very large swarms. At times you have to walk through, the swarms, and there's such a nuisance. Yeah, so that is indeed a mating swarm. Whatever you see is a mating swarm. They're moving up and down. And you probably recognize this area. This is indeed outside the Natural History Building here within the museum. And this is an example of a swarm. And this is the building that houses Nature Kenya right at the back. That's where the upper office of Nature Kenya is. And that's a close-up of a lake fly. Yeah, and you find them so much in the toilets, for instance. If you go to the urinals, if the light were left on at night, because they are also highly attracted to light, yeah, you will find them even inside rooms. So one difference between them again and the mosquitoes is the fact that they also lack a proboscis. That biting and sucking mouthpart, the long, proboscis of a mosquito. Again, their wings tend to cover 3/4 of the bodies, unlike the mosquitoes. So you'd find the wings are up to somewhere there. And then, unlike mosquitoes that will rest with their hind legs up, the lake flies would rest on the walls with their forelegs up. That is the difference. So as you can see, this one is resting on the wall, but the forelegs are raised. They're not touching the wall. A mosquito would have the hind legs over here touching the wall. Being raised away from the wall. Now, we've had so many people, work on mosquitoes, yes, from a long time. Yeah, here in Kenya and globally, and therefore we do have records, some of them quite old records in museums all over the world, and even here within the museum. So our collection is one of the largest of mosquitoes in Africa, and comprises of some of very old specimens. As you can see, this is an example of one of the old. It's probably definitely not the oldest, but I just picked it out of interest. Yeah, this is part of the collection within the National Museums of Kenya. And As you can see, this mosquito was collected in 1922, and a lot seems to have happened in that same year. As you can see, the same year that the very first tusker was made here in Nairobi. So a lot seems to have happened within that year. Yeah, why is this one of interest, this particular species of interest? It's indeed an Anopheles. Yeah, we'll be talking about the various genera. It's an Anopheles, and as you can see, the collector scribbled something here, which you can't see well, but this is supposed to be funesters. Now, Anopheles funesters, for those not in the know, is one of the principal vectors of malaria in Africa. It's such a tiny mosquito, but the damage it causes is not as tiny. Now, what is of interest here is that this mosquito was collected inside the jail. Yeah, there are about 10 of them in our collection, and all of them were collected inside the jail. This is supposed to be Jail. Yeah, so I cropped it, but it's supposed to be Morera jail. Now, when I looked at the records, I didn't find any jail in Kenya going by that name, Morera jail. But then there's a Morera area in Ruiru. Yeah, for those who know Ruiru well, there's Morera. Yeah, Morera just means in Kikuyu, I think it's acacia. Yeah, acacia. Yeah, there must have been lots of acacias there. But then this Ruiru jail is probably the jail that changed name to Ruiru. It probably used to be. I'm yet to go into the historical records and find out whether the jail changed name to the modern day Ruiru jail. So the mosquitoes were collected inside the jail. They are malaria vectors. They were all blood fed. And quite long time ago, 1922, I think they're now 100 years. Yeah, because we are in 2022. Yeah, they must be 100 years now. Now, this is of importance because one, it gives evidence that the malaria vectors were this close to Nairobi. We may not probably have that species now because of the expansion of the city and the disappearance of their preferred habitats and all these. Probably Anopheles funesters may not be there today, but this is a good indicator that they used to be there, this close to the city. Now, the other thing, as I was being introduced, Richard said I have lots of interest in forensic entomology, and this mosquito or these specimens that are here with the missions are of forensic interest to me, because one question I ask myself when I see such a sample full of blood is there a case that remains unresolved since 1922 that this mosquito can, resolve? Yeah. Do we have answers that we can get out of this mosquito that has been lying in a museum collection for this many number of years? And probably there's a case it can solve that remains unresolved up to today. Yeah, imagine somebody, just to give you an example of what I mean, somebody who was implicated of having done, a crime. Yeah, in the year probably 1930. Yeah, or 1925. Yeah, but this mosquito can prove that that person may not have done that or committed that crime because he was not a free man or a woman. Yeah, why? Because he was behind the bars. Yeah, the DNA, from this person is still probably, preserved in the blood that is within this mosquito. Yeah, and if he was behind bars, there's no way he may have committed this other crime that you're claiming that he may have committed. So when I look at such samples, I see them at times with some forensic lenses, and I become very excited. Yeah, so the other interesting bit is that we don't know who the mosquitoes had bitten, whether it was the warden, in those days, the colonial wardens, or whether it had bitten the inmates, we do not know. But if, assuming it had bitten the warden, and the warden was intoxicated with the very first tusker in Nairobi, probably these specimens still hold within them the very first copy of Tusker, to be made in Nairobi up to today. Probably in their berries, they still have that original Tusker from 1922. So quite interesting. I promised to talk generally about the mosquitoes, and I give you just a brief, information about them. Unknown to most of you, we have more than 3,500 species of mosquitoes. This is not in Kenya or Africa, but globally. Yeah, I know that is surprising because to most people who talk to me, they say, I thought they are all the same. Yeah, I thought at most we have only two types, two species of mosquitoes. No, there are that many, and over 3,000 of them are what we call curicines. I will explain it in a few minutes what curicines are, and then about 430 of them will be anopheles. I will explain again what anopheles are. Now in Kenya, we have the major general mosquitoes because they don't belong to one genus, they belong to many different genera, and that is a plural of genus. We don't say genuses, yeah, we say genera. And these are some of the common genera that we find in Kenya and indeed in much of Africa. Yeah, the good old Anopheles, yeah, that is in the mouth of everyone, probably because of malaria. It is there. We've got Culex, we got Aedes, Mansonia. It's quite common, especially if you've been to Lake Naivasha on the shore or the lake. The mosquito that is such a nuisance, that's Mansonia. You got Malaria, Erythemapoditis, and so many other genera. Yeah, so this particular one here of interest is this genus we call Toxorhynchitis. Now, Toxorhynchitis are huge, gigantic mosquitoes, elephant mosquitoes they are called. And one characteristic feature about this mosquito is that it's got a curved proboscis. I did not carve this. It is naturally carved. Yeah, and this mosquito is non-biting. It does not require a blood meal. We see why some mosquitoes, not all of them, why some mosquitoes require a blood meal. A good number of them require a blood meal. But this particular one, the members of the genus Toxorhynchitis do not require blood meal. They do not notice you. And what is more interesting is that the larvae of that elephant mosquitoes exclusively feed on larvae of other mosquitoes. So woe unto you if you're breeding mosquitoes and in your colony of mosquitoes, you by mistake introduce the larvae of the Toxorhynchitis without knowing that they are different because they also look like the other larvae of the mosquitoes. So they will feed on your larvae in large numbers, yeah, in very large numbers. So if you're breeding this mosquito, you must rear other mosquitoes to feed it on them, otherwise it will starve to death. Yeah, even if you try to give it to other creatures, it will not, yeah, it would prefer feeding on mosquito larvae. So this is one of the species that is helping us biologically to reduce the number of mosquitoes in nature. Now, mosquitoes appear in all kind of habitats. You find them in all sorts of habitats, but generally you find them preferring standing water or water that is moving very, very slowly. So swift flowing waters are not suitable for mosquitoes. So that's why you find them being a problem or a nuisance in lakes. In swamps. Swamps are particularly very productive in pools. Yeah, cat traps, in hoof prints, especially the malaria vectors. They so much like hoof prints from animals, buffaloes, cows, and what have you. And then tree holes, especially the genus Aedes. Yeah, that black and white zebra-like mosquito biting you very early in the morning in the offices and late in the evening. It prefers breeding in tree holes when it rains and the tree holes collect water. That's a perfect example, example of breeding habitat for the Aedes. Also the leaf axils, especially plants like bananas, and those of the genus Pandanus and all these. Man-made containers, the tins that you let collect rainwater, the car tires that are lying unused in a compound. So all these are breeding, habitats, and especially again for Aedes. mosquitoes. So this is just simple mosquito classification. Yeah, we talk about brief taxonomy of these mosquitoes. Mosquitoes, like I say, just to repeat for the benefit of those who joined us late, they belong to an insect order or grouping that we call Diptera. Now, these are the true flies, and they all belong to one family, which we call Curicidae. Yeah, so they belong, all mosquitoes all over the world. The 35 plus ones that I mentioned, they all belong to a family of flies that we call Curicidae, which is further subdivided into two subfamilies. We've got Anopheles and Curicidae. Now, Anopheles are the Anopheles or Anopheles, this is a subfamily, and in English, we tend to call them Anopheles. Yeah, Anopheles. Yeah, instead of saying Anopheles, Curicidae, we just tend to call it in English Curicines. So when moving forward, you hear me mention Curicines, just know these are all members of the subfamily Curicidae. And this is how you tell the difference between subfamilies and families. If you see anything adding with ni or ni, that's a subfamily. Anything adding with di or di, it's a family. So that's the difference. So moving forward, I'll keep mentioning Anopheles to mean all members of the subfamily Anopheles, and Curicines to refer to all members of the subfamily Curicines. Yeah, in Africa, the only genus that belongs to the subfamily Anopheles is Anopheles. Yeah. So all the other genera that I mentioned, Eretmapoditis, Mansonia, Culex, Coculitidia, Idiomya, all the other genera that I mentioned belong to the subfamily Curicidae. So they are Curicines. Yeah, but is Anopheles the only genus of the subfamily within in the world? No. It only happens to be the only one represented in Africa. We have two more. Yeah, we have two more, but they're outside Africa, probably not of interest to us. We got Bironera from Australian region and Chagracia from South America. Yeah, so we've got two more. Yeah, but they're not of much interest to us here in Africa. So that's just to correct the impression that the subfamily Anopheles has got only one genus. No, there are two more, only that they are outside our continent. Now, I just want to tell you the differences between males of mosquitoes and females. Very easy, just like human beings. The males of mosquitoes have got beards, if you like, yeah, on the antennae. These are the structures we call antennae. These are the antennae on the female. And if you look at the antennae of the males, they are bushy. Yeah, in entomology or in science, we say they are plumose. Yeah, but it simply means they are bushy. Yeah, if you look at the antennae, same antennae, but on the females, they're simple, what we describe as simple. Yeah, they are not as bushy. Yeah, we got several other differences. So this is just a continuation of the differences. We just talked about the bushiness of the antennae. As you can see, these are males and those are females. They're not as bushy, so don't confuse this. Yeah, doesn't mean they don't have hairs. They got simple hairs, yeah, but they are not as bushy as the males. Then this is a biting mouth part, which I call the proboscis. If you look on both sides of the proboscis, the biting mouth part, we've got two structures, which we call the palps. Yeah, and we have got in this one as well. Yeah, in some, the palps are very short and others are very long, especially in Anopheles, they're very long. But if you look at the palps, they are not bushy and they are not swollen at the tip. Those are females. But then if you look at the same palps, in the males, they are equally bushy, just like antennae, and they tend to be club-shaped, swollen, slightly swollen on the tip. So those are some of the things that can help you very quickly identify the differences between the males and the females. Now we've talked about various genera. Yeah, I remember. Now genuses, genera, we talked of Anopheles, we talked of Aedes, Culex, probably the ones that are well known to the audience here. But are there differences between the genera? Yes, there are. And indeed, the differences are in all their stages, starting from the eggs, then the larval stages. The differences go on to the pupae and right to the adults. Now, what are the differences? For instance, this is an egg or these are eggs of Anopheles. Anopheles lay their eggs singly, moja moja, one singly. Whereas most curicines, like I said, I'll be using the word curicines to refer to all these other genera. Most of them lay them in rafts. We call them egg rafts. They are packaged like, sausage. Packages, the way you buy your sausages, as you can see. So these are times hundreds of eggs, in one raft. Aedes tend to lay there singly, just like Anopheles, but there's a difference. If you look at the eggs of Anopheles, they will have some floats on the side. They have something to help them remain, floating on the surface of the waters. They're like buoys. So the next stage is the larval stage, and the larvae of Anopheles lie flat on the surface of the water. They do not have what we call a breathing siphon. So to acquire oxygen from the atmosphere, they have to lie flat on the surface of the water and be in touch, in contact with the air in the atmosphere. So that's why they lie flat on the surface of the water. Whereas the larvae of curicines, non-Anopheles now, all the curicines, they lie at an angle, into the water. They have the advantage of having a breathing siphon, so they can access, air or oxygen from whatever angle or position they lie in. They don't have a problem like Anopheles. How about the pupal stage? This is the pupal stage. The pupae of Anopheles have got the breathing trumpet. Now the siphon, when they turn into pupae, this breathing siphon turns into a trumpet. Even the name changes. And as the name suggests, this trumpet will be widening. It will be widening outside. It will start as narrow and then it will widen as you go towards the surface of the water. Whereas all the curicines, their trumpet is cylindrical, like a water pipe. All the, it's of equal dimensions. It's cylindrical, as you can see. This one tends to broaden as it moves out. How about the adults? Probably this is the only difference most of you know. You probably know the differences in the other stages. The adults of Anopheles, and this is how to tell Anopheles next time you see one on the wall, is that they tend to start at an angle with their abdomen raised. You remember the difference I gave, just as a recap, between the lake flies and the mosquitoes? The fact that mosquitoes rest with their hind legs raised, whether they are curicines of Anopheles or their hind legs will be raised, whereas the lake flies would raise their front legs. So the Anopheles will start at an angle, sometimes almost 90 degrees for some species, whereas all the curicines, they rest parallel to the surface on which they are resting. I'm sure with this kind of information tomorrow or when you meet, you can be able to tell me whether you've got curicines or you've got Anopheles in your room. So this is just a close-up of the Anopheles eggs. Remember, I say they have floats, floaters or buoys that help them float on the surface of the waters. It's just this structure on each side of the egg that is full of air. And this is just a close-up. So Aedes eggs, although they are laid singly, just like Anopheles, they do not have the floats. So that is one difference between them. This is a simple life cycle of a mosquito. And mosquitoes and indeed other flies, they follow what we call a complete metamorphosis. Yeah, and this is a life cycle that starts with an adult laying eggs, and then the eggs will hatch into larvae, and then the larvae will turn into a stage, which we call the pupae. And then, of course, the pupae will emerge, the adult will emerge, a winged adult will emerge out of the pupae. Now that is complete metamorphosis. Yeah, you know, other insects that follow incomplete metamorphosis. Yeah, now incomplete would mean they lack the stage, this pupa stage. So you'd have the creatures hatch from the eggs into what we call nymphs. And the nymphs are almost like the adults, only that they don't have wings, and they keep on growing bigger and bigger as they develop the wings. And finally, they just turn into winged adults. A very good example are the grasshoppers and locusts. So you have the grasshoppers laying eggs, which will hatch into nymphs. And those nymphs will go through what we call the nymphal instars, various nymphal stages as they get bigger and bigger, and eventually they get into a locust, a winged grasshopper or locust. They do not have the pupa stage, and therefore that is incomplete metamorphosis. Not so for mosquitoes. This is a complete metamorphosis. Now, what would be the role of mosquitoes? Probably most of you think none. Yeah, they are good for nothing. Yeah, they all should be sprayed and killed. Yeah, but I tend to disagree. Unknown to you is that mosquitoes are good pollinators. Now, if you went to the flowers at night, you probably didn't have any good reason to go to watch flowers at night. But now, from this lecture starting now, if you happen to go to the flowers and you keenly look, you know, with your torch, you might come across so many mosquitoes on the flowers. Yeah, most of them at night. If you're lucky, you see a few during the day, especially the black and white one, Aedes aegypti. Also the big one, the elephant one I showed you, the Toxophytes, they'll be feeding even during the day. Now, what are they doing on the flowers? They are looking for nectar because this is their source of energy. They depend on that for their energy. So they get nectar from the flowers. And in the process, of course, just like bees do. Yeah, bees are not going to pollinate your flowers. No, they're looking for nectar. They're looking for pollen. But in the process, they do the pollination. So it's not any different with the mosquitoes. So next time, be keen towards evening, maybe not very late into the night, you'll be able to see mosquitoes foraging for nectar, and in the process doing the pollination. They are good source of food for higher life. This is a mosquito being fed by fish, and indeed lots of fish feed on mosquitoes. You probably have heard of the mosquito fish, the guppies. Yeah, they feed on mosquitoes in large numbers, thereby helping reduce the numbers. And this is a dragonfly, indeed, with a mosquito in its mouth. Yeah, it's almost munching the wings and the legs. Now, mosquitoes are also very good indicators, and particularly of a changing climate. We do have mosquitoes that you find in extremely cold places. And here in Kenya, I'll give an example of a mosquito we call Anopheles. You probably now, by now know the genus Anopheles. Anopheles christiae. This is not a malaria vector, actually. It doesn't bite humans, prefers sheep. And you find it in very cold places, extremely cold places. And if you go to the collection in a Natural History Museum, you'd find places such as Kericho, you know, places that are synonymous with coldness. You find places, specimens from places like Meru, Limuru, the Aberdares. Yeah, so this is a cold place mosquito. Now, what is happening with the warming up of these areas? They are retreating, you know, upwards towards the mountains, you know, where the temperatures are still cold. And if you have the baseline data where they were in a particular year, you can actually keep on monitoring how the environment is warming up just by just looking at some of these creatures. And the vice versa is true. There's some, the reverse is true. There's some species, especially the malaria vectors like Anopheles finesse, as I mentioned, Anopheles gambi, another principal vector of malaria that you find in extremely hot and humid places. That's why it is a problem down at the coast. That's why it's a problem in the lake region. Lake region, meaning Lake Victoria region. You get there and you feel this humidity and very high temperatures. Now, what happens to those species when the highlands, the high altitude areas start warming up? Yeah, we might start seeing them encroaching up, the mountains. So it is good to know where they are at the moment, so that 100 years time, to come, we know how they have changed, how their distribution has changed, and that will be a measure, an indicator of changing climate, warming climate. Yeah, so they are very good in indicating a changing climate. Yeah, probably this is all that most people listening relate, to malaria disease transmission. Yes, indeed, a few species bite humans. They interact with humans, and it is that human-mosquito interaction that is of importance when it comes to disease transmission. Yeah, a mosquito that doesn't notice you, doesn't require blood meal, like I gave a few examples. Yeah, it is not of any importance when it comes to disease transmission. It is indeed that human-mosquito interaction that is key. So a few species are able to do this. They interacted with man in that kind of manner, and therefore they are known disease vectors. So this is Elephantiasis, quite common along the coastal areas in Africa and in other parts of the world, in Asia and other parts of the world. And this is the good old malaria, affecting lots of people. So a few species indeed, spoil the name of otherwise good creatures that are mosquitoes, good creatures that are pollinators. They are indicators of climate change. They are food fish and dragonflies and what have you. So a few species will spoil their otherwise good name. So how do we collect these creatures? We've got lots of methods and we've got lots of tools that are available for collecting mosquitoes. And just to mention a few of them, this is a structure, very simple thing we call an aspirator or a putter. Some people call it a putter, and this is how you use it by just sucking your specimens. When you spot them on the walls, on trees, on vegetation, you just suck them with your mouth into a putter. It's got a screen here over here to prevent whatever you're sucking going all through into your mouth. At times it happens if it is torn, so you must check that your screen is in good shape. Otherwise, you might you might have blood-fed mosquitoes into your mouth. So this is a gravity trap, we call it, and it targets mosquitoes that are about to lay eggs. They are full of eggs and they are looking for somewhere to lay. Not all of them, but particularly Culex, especially the urban problem, that is Culex quinquefasciatus. Yeah, in some regions it's called fatigans. So this is the nuisance that we have in most urban areas in the world. Nairobi particularly is a big problem. And the reason why it's a big problem is because it thrives, it breathes in organically enriched waters that we have from kitchen waste, from sewer lines and all this. That's why it's a big problem in urban areas. So basically what you do is you try to replicate their egg-laying habitats. The sewage water, the gray water from kitchen and water view is what attracts this mosquito to laying eggs into that kind of water. So basically we try to mimic that water, but we don't use sewage in our case. We just let some leaves, particularly mango tree, mango leaves, rot in the water for several days until it stinks. And then you just pour it into the basin here. There will be a fan sucking upwards, and all the mosquitoes that are coming to look for this, very nice habitat for laying their eggs, they are sucked by the running fan into the collecting net. So the white thing is a collecting net. And at times you find them in their thousands in the morning. Females, because it will be females that will be full of eggs and they'll be looking for somewhere. A few confused males get there. I don't know, probably they'll be falling females. I don't know why they get there as far, but you find a few confused males. And this is what we call the BG Sentinel Trap, and it targets mainly IADs. Yeah, because some lure that is put somewhere down beneath this netting material is full of pores, and then the fan is blowing outwards, to get the chemicals into the environment, and another fan is blowing inwards. Yeah, it's basically one fan that is blowing inwards, but that air also comes outward, distributing distributing the lure or whatever bait is put in this kind of trap. So this, like I say, targets mainly the Aedes, the genus Aedes, and particularly Aedes aegypti, which is a well-known vector of so many viral diseases: yellow fever, chikungunya, dengue fever, and so many others. Now, this is a good old light trap. We call it the Cdc Light Drop, and uses a light, from a bulb here. And mosquitoes, as you know, are highly attracted to light. Yeah, but you can also bait, add this CO2 bait to it. And when you add CO2 to this light drop, at times the catch is increased tenfold. Yeah, so yeah, but even if you don't use a CO2, mosquitoes still be attracted to this light that is coming from this bulb over here. So there'll be a fan, of course, running downwards, and everything that will be attracted to the light will be sucked into the collecting net. Yeah, so the major mosquito attractants, of course, will be carbon dioxide. If you did not know, whatever you exhale, will be part of what attracts mosquitoes. And then also, fatty acids and also byproducts from the body like ammonia, lactic acid, octanol, and of course, heat. Yeah, carbon dioxide is particularly very important. And it's a long-range attractant. This is probably what will bring mosquitoes from very far away from the breeding site. And when they get near to you, other, attractants take over, like ammonia, lactic acid. Yeah, heat around you also increases. It's able to pinpoint where you are because, of course, heat around you also increases. So CO2 is presented either as gaseous form or dry ice. These are ice pellets from CO2, of course. When CO2 is compressed, at a very high pressure, you get dry ice, and you can also present it in this kind of form. In Kenya, we mine CO2. Yeah, you probably know of carbon dioxide mining up in Kirita Forest. So next time you see a big, truck looking like that of LPG, written carbaceid on it, just know it's transporting CO2 from the mines within Kirita Forest to the factory in industrial area. That's why we buy our CO2 quite cheaply. And for the environmentalists who worry about us creating more CO2 and the global warming and all these, this is not, made. It is coming from the mines. It is coming from underground. It is only mined and put into containers. So this is just making one more use of this CO2 before it, disappears into the environment. I would equate it to water in a river, in River Tana, for instance. If you don't use that water to generate electricity, it will still go to the ocean. So producing electricity, it's just making one more use of that water before it disappears into the ocean. So this is basically what we are doing, making use of that CO2. We are not generating it. We're just using what is coming from the mines. So why rear or breed mosquitoes? Yeah, that's a weird thing, isn't it? So we'll be discussing next why some people are this much weird. And one reason is for the ease of identification. It's easier to identify adults as opposed to identifying other stages, identifying them from eggs or pupae or larvae. It is much easier if you have an adult, a winged adult in good shape. So some people would rear them for that reason. And then when you want to develop a reference collection like we do have here in the natural history, museum, bit of the Museum of Kenya. So you rather rear your specimens from the larvae or all the way from the eggs. You hatch them, let them go through the larval stages, they pupate, and then the adults will emerge, or whatever stage immature stage you find them in, because you might find them in as larvae, you might find them as pupae. Fine. If you let them emerge, if you rear them, you breed them and let them emerge, then you have intact specimens, for your reference collection, and also will ease identification. Mosquito features easily come off. Yeah, if you look at most mosquitoes, you find they have like white spots. Actually, the white markings that you see on what people call the zebra mosquito are basically scales. Yeah, and they're scales like those on the fish. So these scales very easily come off. So they're basically black and white scales. That's why it appears like a zebra, and they very easily come off. And they also have tiny hairs, which we call setae, that very easily come off. So if you collected your mosquito, say through sweeping, using a sweep net, you can imagine the beating they all get with everything accumulating in the net, with the debris, with the vegetation and other hardy insects like beetles and what have you. So you have a total mess, and you will not have the key features needed for identification retained. So to make them have all the features, it will be good to rear your mosquitoes. Yeah, so at times we need constant supply for various applications in research. And a few examples I have highlighted there for research. For instance, if you want to do some research on insecticide resistance, if you suspect that the mosquitoes are resisting this kind of chemical or the other, you will therefore need mosquitoes, which will be subjected to the chemical you suspect is being resisted. So there's no way you can test for resistance without using live mosquitoes. Yeah, if a company, and they're there, and there are many, has got a new bed net, that is impregnated with chemicals, pesticides, and it's supposed to do ABCD, yeah, you need to test that claim that this bed net is capable of doing ABCD. So you need to test the efficacy, of that net. And to do this, you definitely need to subject it to live mosquitoes of different species or of target species. The same with new insecticides. If you come up with a new molecule, a new chemical that is supposed to target mosquitoes, then it goes without saying that you need mosquitoes to test that new molecule. Yeah, how about the transmission of diseases? There's so many research institutions studying the transmission of the diseases. Yeah, and all other techniques like, male sterilization, genetically modifying a little bit the mosquitoes in an attempt to control the wild populations. And this particular one requires lots, thousands, hundreds of thousands of mosquitoes. So these are some of the reasons why some people maintain colonies, and they have maintained them for centuries, yeah, and decades. So how do we do this? Yeah, of course, we rear them in special rooms, which we call insectaries. Yeah, and that's a good example of an insectary. An insectary can be simple. They range from very simple to very high-tech, modern insectaries. But like I say, they can be very simple. And basically what you're doing within that insectary, you are subjecting the mosquitoes to the right environment, the conducive conditions that they need for growth. So for instance, the temperatures for breeding larvae, especially the Anopheles, will be between 32 to 34. And for adults will be much slightly lower, 26 to 28. And then the relative humidity, remember, you don't want them to desiccate, by subjecting them to very, very low humidity levels. So you would maintain your humidity within that range. And that's why when you land from an aircraft in Mombasa, you feel this humidity hitting you. Yeah, that's the kind of humidity that mosquitoes need to survive. So you're trying to mimic that within an insectary. Another very important thing is regulating the photoperiods. Yeah, now photoperiods, especially our mosquitoes, are accustomed to 12 hours of light and 12 hours of darkness. And you give them something else out of that, you are likely to get problems. They may develop normally, but then you will have trouble when you want them to lay eggs. They may just fail to lay eggs simply because the photoperiod was not well regulated. So we regulate all these. And here in Africa, we just subject them, since we don't have the winters and what have you, when the times changes and all this. Our times are 12 hours of light and 12 hours of darkness within the equator. So yeah, so for mosquitoes to lay eggs, they need a blood meal. Not all of them, yeah, but all those that you see biting, yeah, either biting you or cows or chicken or snakes or what have you, amphibians, then it means those are the species that require a blood meal. Not all of them. I want to emphasize this. There's some that lay eggs without a blood meal. But why do they need a blood meal? Yeah, is blood food for mosquitoes? Because this is what most people, ask. Is blood food for mosquitoes? And I tell them no. What is food for mosquitoes? And we already talked about it. It's nectar. This is where they get their energy from. But in addition to that, the females of the biting species require some proteins for the maturation of their eggs. And that's why they basically come for a blood meal. So when next time you see a mosquito biting, you just know one thing straight away, that it is a female. Yeah, so the other thing you should know is that it's just about to, have its egg, or it wants to have its egg, mature. It's probably just being fertilized, and therefore it wants the eggs to mature. That's why it's coming for the blood, for the proteins that are found in the blood. And the blood, because given their mouth, design, it's like a pipe, a very long needle, yeah, that cannot get protein in any other form. If they had chewing mandibles, maybe they would go for roast meat, nyama choma, if all they want is proteins. But then they can't, because the structure of their mouth part, their protein source has to be liquid in nature. So at times we provide our own, blood that somebody whose hand is inside the cage. And as you can see, we got all these females. Yeah, not even a single male, all these females taking a blood meal. Yeah, one caution about this is that you must never do this with a wild population, mosquitoes that you have brought from the wild, because some of them come with viruses right from the wild. They're capable of acquiring viruses even from eggs, something we call trans-ovarian transmission, whereby the viruses will go through the mother to the eggs, and the larvae that hatch out of those eggs will be automatically infected, and the pupae that pupate out of those larvae will automatically be infested with the virus, and the adults that emerge will automatically have the virus. So if they bite, they are likely to transmit the disease. Now, what we do, the instances when we only do this kind of a thing is when you have a clean colony that has been in captivity for centuries, for decades, and it's been screened and it doesn't have it. It's known not to carry any infection. And even then, if you feed it, only you can feed, the next time or the next generation and all this. Because now if you share, then you are likely to transmit disease, not from the mosquito now, but now from the people who are sharing the feeding. Fine, some people do this, some labs do that, but there is this nice alternative here whereby we feed them through what we call membranes. Yeah, so this is membrane feeding. This comes as a complete structure, and you source your blood from wherever, from slaughterhouses and all this, and you subject your blood to the mosquitoes through this structure, through the membrane. And as you can see, they very comfortably bite through the membrane, and they get and gauge. Some people use laboratory animals. I particularly don't like it. Yeah, you know, the guinea pigs and what have you, but I particularly don't like it. So after feeding, the mosquitoes will rest. Yeah, they'll actually go into dark places and rest. That's why at times when you pick coats, you know, hung from a wall, you just see so many blood-fed mosquitoes flying away. They were just resting, waiting for the blood to digest and the formation of the eggs. Yeah. So at times they suck, in most cases, they suck until some liquid appearing like blood oozes out from their behind. Now this is sort of like the plasma, you know, the clear water in the blood is like plasma, and they do this to concentrate, you know, the blood cells, and hence the proteins in their system. So they are like trying to concentrate the proteins by letting the plasma ooze out from their behind. So this is not uncommon if you watch a mosquito you know, bite you. And that's why in the caricature, right at the first page, you saw some blood on the floor. It's not that the mosquitoes were so drunk they could stagger to the bayou rhino. No, it is a common thing when the mosquitoes do the feeding. Now, these are the various stages that the blood go through after feeding. As you can see, this is an unfed, hungry, very hungry mosquito. And then when it's fully fed, can actually see the blood is just like in your system, and then it will keep on darkening as it gets digested. And as the digestion happens, right from the tip, you see the abdomen whitening. Now this part is where the eggs have already formed, and as the blood disappears, of course, as the digestion happens, you keep on getting the whitening, meaning the eggs keep on forming until it is all white. And at this stage, we say it is fully gravid. Yeah, it is just looking and waiting to lay the eggs. So in captivity, what we do, we provide them with filter papers that are damp. Yeah, just damp filter papers. Imagine they sense that dampness and they take that to mean a habitat full of water. They will imagine it is you know, the shores of a swamp or lake, and they will deposit their eggs onto the wet filter paper. These are eggs on a filter paper. And therefore you collect your eggs, and if you want to continue with your colony, which most of us do, you just flood the eggs into a tray with water. And within a few days, depending on species, they will hatch into tiny larvae. Yeah, you see them wriggling on the surface of the water, and then they get bigger and bigger, and they will pupate. And when they get to that stage of pupae, you must harvest them, because if you don't, the following moment your insectary will be full of flying mosquitoes. And you do not want to do this, particularly if you're dealing with dangerous species, yeah, like the malaria vectors or the yellow fever vectors. So there has to be somebody who notices the pupation, and they will collect all the pupae and pull them into the cages. Yeah, this enclosure. So they cannot get out of this cage now. So the emerging adults will be enclosed in here, and the process can repeat. This is how we maintain mosquitoes, and some laboratories globally have maintained them for many, many decades, yeah, if not centuries. Yeah, so that is how we maintain. This is just a close-up of a container breeding. It's indeed here at the museum, container breeding, one of the malaria vectors. And this is Anopheles funestus, as you can see the name. And if you remember me describing Anopheles, I told you the larvae lie flat on the surface of the water. So you should be able to tell me that indeed those are Anopheles larvae. Now, there are opportunities of applying innovation and technology, and I like mentioning some of these things, especially when talking to young people, young minds, young innovators. Now, this is my breeding room within here, the museum, somewhere down where I'm seated. It's a simple breeding room, and this was during the the COVID outbreak. This is when I was wearing mosquitoes. And what happened was because, as you know, there was reduced presence in workplaces. People are encouraged to stay at home and to social distance, to reduce unnecessary movements and contacts. But then this came with a problem for people who had, say, like colonies, and they had to attend to them. But we also felt like we also need to be away, just like everybody else. As essential as this one is, because indeed this can be and was classified as an essential service, we also felt that it's good to reduce the risk. So what we did, or what I did, I came up with this idea of automating the rearing of mosquitoes. And therefore myself, together with a few tech people, we designed a robot that could do everything that a human was doing before the COVID came. So one of the things that the robot was supposed to do is indeed the photoperiod regulation. Subjecting our mosquito to 12 hours of darkness, whether I'm there or not, whether I'm staying at home or not, and 12 hours of darkness. So that is one of the things that the robot that we designed was supposed to do. And it did, as you can see, by 5:30 p.m., the room is very well lit, and it will start dimming by 6 p.m. So that's the thing. You don't put the switch off like you do when you're going to bed for mosquitoes. You must mimic, daylight and darkness and dawn and dusk. So, and by 6 it will start dimming, and by 6 it will get even darker, and by 7 p.m. it will be totally dark within the insectary. And then the following day in the morning, by say 5:30, the reverse would happen. It will switch on, but gradually get brighter and brighter, and by say 7 a.m., the room will be fully lit, whether I am there or not. And this has saved a lot of time. Because mosquito rearing is indeed a very time-consuming, exercise, and it has saved us a lot of time because we don't have to carry some of these functions, and because of that, we are even able to go to the field. To go to the field, you just lock your insectary, knowing that the mosquitoes are well taken care of, and you just go. But who feeds them? That's the next question. So the robot is supposed to do the function of the feeding as well. And the feeding is staggered. You don't feed the first instance, the very young instance, the same way you'd feed the fourth instance that are just about to turn into pupae. During that time, when the fourth instance, they need to take a lot of food, consuming a lot of food, and much of it is not spending a lot of time on the water. Otherwise, if you have too much food remaining on the surface of the water, you risk scumming of the water, which is as a result of a bacterial activity, and you are likely to have mass mortalities of your mosquitoes because it means they will not be able to access the oxygen. It's like pouring paraffin onto the surface of the water. So if you have a scum, that's a terrible thing. So to avoid this, especially when they're young and they're not consuming all this food very easily, you sort of feed them after a very long duration. So you feed them maybe only once after 3 hours. Then after another 3 hours, the robot will feed them again. After another 3 hours, it will do that. But then when they increase in instance and the requirement, the energy requirement also increases, you have to reduce that frequency. You have to increase the frequency, you have to reduce the duration of waiting. So it will maybe feed them when they're second instance every 1 hour. Every 1 hour. When they're fourth instance, maybe every 20 minutes. All this is within the program or the robot. So we just program it and it will know that they're in day 1. So this is the frequency. We are in day 2 and this is the frequency. We are in day 8 or 7, and this is the frequency. Yeah, so you don't have to be there. So you're probably wondering how the robot does the feeding. I will demonstrate to you in a way of video. And so besides the writing, as you can see, which is very bright, this was during the day, it had not started dimming. And that is now how the robot does the feeding, and it's capable to feed so many containers at the same time. So meaning you can rear so many numbers at the same time, even if you're not there. Yeah, so these are some of the opportunities, even within entomology, within mosquitoes, for innovation. And what I tell young people is that you don't have to reinvent the wheel. Yeah, almost everything has been, discovered. There are few things remaining to be discovered, don't get me wrong, but quite a number of things have been discovered. And what I mean is that electricity is there. You don't have to reinvent electricity. The motors are there. You don't have to make another motor. Yeah, the chips are there. The computer chips are there. You don't have maybe to remake another chip. But what now remains of us is to get this part, another part that is already made, another part that is already made, bring them together, make a product that carries out a function, as intended. Yeah, that's basically how an aeroplane, Boeing, 787 moves. Yeah, it is this part and another part and another part put together, and it makes an airplane that is such a marvelous innovation. So this is just to challenge the young people, because when you hear of innovation and technology, you tend to think it's such a big thing. It isn't. Yeah, what I have just explained is that you just need to get what other people have made, put them together, come up with a product like I have, come up with one, and it is carrying a function as I want it to do. So that is the end of the talk, and I hope you enjoyed it. I anticipate some questions, which I believe you've been putting in the chat, and Richard, hopefully you've been kindly noting the questions for me. Yeah, so thank you very much. And I will stop sharing. Or probably just leave it there just in case I need to go to a particular slide.
Okay, thank you very much, Mr. Laban, for that very informative and educative talk. In fact, I have really learned so much about mosquitoes. In the chat box, I see some comments in here from John and Margaret Cooper.
Yes.
Thank you.
Of course.
Yeah. Okay, thank you, Laban, for your fascinating lecture. Nzuri sana. When we traveled to Lake Tanganyika, we saw plumes of lake flies rising from the lake. The local people got the flies on greased plates and made cakes that tasted like fish. When the lake flies came to National Museums of Kenya, did you make a cake?
I don't make cakes to begin with. Yeah, first of all, thank you. Yeah, nice to hear your voice. At times when you're presenting, you don't know whether you lost everyone. You're probably talking to yourself and you don't know. Yeah, at times the net misbehaves. So I'm glad to hear people talk, and I know, oh, we've been together all along. So thank you very much. And thanks, John and Margaret, for the question. To begin with, I do not make cakes out of the lake flies. I do know there's species that are edible, and in fact here in Kenya we do have them being eaten in the lake region. The communities around the lake, particularly the Luo community, call them Sam, if I get it corrected. That's the right way of saying it. Sam, you prolong it a little bit. This is the species that is edible, and it's it periodically comes, not always, it periodically comes from the lake towards the villages, just like you witnessed in Lake Tanganyika, and I tend to believe it's the same species. So when the fishermen in the lake see some coming towards the villages. And in the days of mobile phone, they call people at home and they tell them, some bill, some is coming. So the villagers get ready with buckets and whatever they need to harvest, some for dinner. Yes, I know some people are eating them, and they are cakes, indeed also from the lake region here in Kenya. But yes, I don't, to answer your question, I don't make cakes out of them. Talk of preaching, preaching water and taking wine.
Okay, thank you very much, Mr. Laban, for that. I was actually waiting to hear if there are cakes somewhere. From Edwin Muga, thank you, Laban, for your informative presentation. You mentioned about the elephant mosquito and the possibility of its use as a biological control of other species through.
Indeed, yeah.
Is there currently such a program being implemented in this regard?
No, I think I don't think so, because they are probably better, in nature. They thrive so well and they multiply so well in nature. Yeah, the trouble is the condition in which the habitat in which they are found in nature. At times it's very hard to replicate this. Yeah, it's not easy to replicate every kind of condition and habitat within an insectary. Actually, up to now, there are some species that of importance because they're disease vectors, but you are unable to rear them in captivity because of the same reason I have explained. But in nature, they just thrive. Yeah, so we just let them do the control in nature where they are able to do it without any problem. Yeah, so at the moment, I don't know of any attempt of rearing the elephant mosquito, the toxolychitis, to do this. But in nature, they just do it. So we only need to conserve the habitats in nature, whatever they like, not tamper with it, don't pollute it with chemicals, don't drain them and all this, and they will do the working, for you, free of charge, by the way. I normally tell people, if on average you had 10 bites of mosquitoes in one evening, these bites would have been 100, were it not for such creatures that help you bring the numbers, and their effort go unappreciated, unnoticed, so the bites would have been hundreds or thousands, were it not for dragonflies, were it not for the elephant mosquito and what have you.
Okay, thank you very much for that insightful response. From our Facebook page, I have some of our followers, like Philip Shamir saying what a learning. Philip Shamir is from Naivasha. And there's a question from Mude.
I miss Naivasha.
Yes, from Naivasha.
Yeah, I must go there soon.
Oh, great. You will meet Philip Shamir there. Also from Patrick Mude, he's asking through our Facebook page, is there people or blood that is more preferred by the mosquitoes?
Wow. Yes, actually there was a study or an article not too long ago, just this week, that came out, and it showed, they sort of proved that the blood group B, is relished, by mosquitoes. Doesn't mean it's the only one, but they tend to have a higher liking for anyone with blood group B. That is to answer whoever asked that question. But besides the blood group, which in my opinion probably isn't a major factor, we do have, some people producing more of one attractant or the other than other people. I just talked about what attracts mosquitoes to us. Remember the mentioned lactic acid, ammonia, octanol, CO2. So these are the chemicals that we produce, and some people produce more than others, and therefore they are more attractive than others. People who have, for instance, smelly legs, smelly socks are more relished by mosquitoes than people who tend to keep their feet clean. Yeah, because in the sweat, you know, in your toes and all these, in the dirty socks, you have lots of lactic acid and you've got lots of ammonia. Now, I want you to notice something next time you go to the bush, and when you've got a lot of the zebra, I'm calling it zebra mosquito so that most people can know exactly what I'm referring to. But this is the Aedes aegypti, the day-biting mosquito that tend to be striped, black and white like a zebra. Yeah, it will tend to land on your shoes, areas near your ankle. That is where they'll be swarming. Yeah, never beyond the knees. Yeah, so it's interested with the lactic acid, with the ammonia, urea, and what have you that you have in the feet coming from in between the toes. So, and the other thing I want you to notice is next time, because it's a common mosquito in houses and in offices. Next time you see it, you might notice that it's not even interested in you. Eventually, it will be interested because you're also attractive, but for some time, it will be flying into all the places you've been touching, to the cup where your tea is, and particularly if it is even hot tea, the better, because you have added an extra thing, heat. But then it will move from the cup to where your mouse, computer mouse is. Yeah, I want you to do this observation next time. It's now raining most places, so they'll be up and running everywhere in a few days. So it's attracted to the lactic acid, to the ammonia that is coming from your sweat, and it is in all the places that you've been touching, particularly the computers, the laptop, the keyboards, the mouse, the cups, and all these. Yeah, eventually it will come to you, but for some time it will be confused, trying to find where all this is coming from. Yeah, so yes, besides the blood group, which I answered, I said yes, because research has shown this. There are other factors that attract mosquitoes more than other people. Yeah, especially if you've got more attractants in the name of lactic acid, ammonia, urea, octanol, CO2. Yes, you'll be more relished than others.
Oh, great. I think Patrick has got sufficient answer there. Margaret Vaz, thanks for sharing such interesting information. Have we lost the balance between the different species of mosquitoes?
The balance?
Yes.
Okay, yeah, I think yes. Yeah, it goes without saying that those that relate with man have gotten the beating, you know, from man, and those that don't notice man and therefore of no importance to him as far as disease transmission is concerned, are safe for now. Yeah, they're not totally safe because when we, for instance, apply chemicals, they are indiscriminate. They don't, discriminate. They don't choose who to kill and who to spare. Yeah, so they are also not safe. But imagine a mosquito that comes indoors every evening, and when it comes, it finds sprayed walls, chemicals. We call it residual sprays. The sprays that at times the government sprays, especially in malaria endemic areas, that's supposed to remain for a long time there. So every time they're coming in, they're not living, they're falling dead. And in addition to that, people have got treated bed nets. So these are targeting only those that are coming indoors. Yeah, and they'll come today, they'll come tomorrow, the day after, next year. So you can imagine what will happen eventually to their numbers. Yeah, they tend to, and that's why you see the government reporting fall in malaria incidences. Yeah, it's basically because the nets and the residual sprays are working and other measures. So eventually there will be an imbalance. Yeah, but then those that are relating with man so much will be more hit than those that don't even notice man. Yeah, but even those that don't notice man are not totally safe because of what I mentioned. Yeah, agricultural pests are also a problem. Yeah, so whether a mosquito comes, indoors or not, it's equally also not 100% safe because we have all these things that we are doing out that are not for them. Yeah, destroying their habitats, And not that I'm advocating. I mean, these are not like dragonflies or elephants. Not that I'm advocating for their conservation. Yeah. So, but all I'm saying is that they are also not safe from all the things that human beings are doing. But they're much better than those that interact with man. Yeah. So yes, there's definitely some imbalance.
Thank you. Compliment from Paul. Thanks for a good talk. And down to Mwesh. Thank you, Laban, for a very informative talk. I have learned so much about mosquitoes. I have a question about the robot. How do you collect data from it? And do you monitor errors in case a feeding was missed?
That's a good question. Mwesh is only 2 meters from where I'm seated, and is a spider lady. And I think that's a very good question, Mwesh, about the robot. Yeah, there isn't data, the way it is designed at the moment, there isn't any data to be collected about it. Yeah, so I would say it is helping me collect data about the mosquitoes. Yeah, because when it assists me in rearing, of course I have, some targets. Yeah, there must be some research that I'm doing. It is that data, it will help me collect. So it's probably the other way around. But at the moment, it doesn't have, data that it logs in that I need to collect. No, but it assists me collect data. Yeah, at times it goes haywire. Yeah, and especially if, say, there's a blackout at night and either the power came back and there was some surge and all these, at times it may misbehave. Yeah, and at times at times I find there was a problem and either it stopped or it did something that it wasn't supposed to be done. So it is work in progress, and we are still looking at how to, solve some of these issues. So it is still work in progress. Yeah. And I would say yes, just to answer you, yes, there's some instances where you find that it did something it wasn't meant to do. Probability overfed them. Yeah, because power came and then the memory was lost at some point. And when power came back, it misbehaved and became too generous to the first instar, loving giving them plates upon plates of the food, and they will overfeed and they will be scammed. So we are constantly checking because it is a product in development until it gets polished. Yeah, we are planning to patent it, and that's why I didn't show you how it works out the whole thing. Any other question?
Okay, thank you. We have a question from Odile Kin. Interesting talk. Thank you, Laban. We lost your voice a few times, but the slides were there. Is there any thoughts of using sterilized males in the environment to reduce malaria affected species?
Is there what? Repeat the question. Is there what?
Is there any thought of using
thought? Okay, I see. Yeah, yeah. I don't think it's a thought anymore. I mean, it's a practice that is already there, has been there. People have tried it, probably not here in Kenya, but other parts of the world. But it's not even a thought. Yeah, it's something that is happening. Yeah, it's happening in Brazil. It's happening in other places. Yeah, probably some parts of Africa. Yeah, sterilization techniques, and yes, it's already there. Yeah, but. Yeah, I don't know why it's never very successful. So you try this. One lecturer once told us when I was at the university that, do you know mosquitoes also have their own university? You try this. They also go to their university and they find a way of countering it. Yeah, so no matter how much you try, they also find a way of countering it. So the techniques are there, they've been there, but whether they have attained much, yeah, I just leave it to you.
Okay, great. A question from Lucy Mwangi, and she's asking, what are your thoughts on genetically modified mosquitoes as is being studied in some spheres in the malaria prevention field?
Yeah, well, definitely I think there's some positive to it, but I'll be very careful. Yeah, since she asked for my thought, I wouldn't do it myself. I have my fears, about this, because it involves changing, an organism. We know what changes can bring, especially in organisms. My take is, and this is where my fear is, maybe they are unfounded. If you genetically change this creature that was transmitting malaria, how sure are you that you don't now make it suitable for other diseases that it couldn't, simply because of its genetic makeup? It couldn't transmit HIV/AIDS simply because of its genetic makeup. Now, if you tamper with that, are there likelihood of now making it suitable, for transmitting other diseases that it couldn't do before? So these are some of the fears that I have, and I'm sure so many other people share the fears. And yes, myself, I don't support that.
Okay, a question from John. Thank you for a fascinating presentation. I'll never look at mosquitoes in the same way again. Am I right in thinking that only Anopheles species act as malaria vectors in Kenya?
You're not just in Kenya, globally. You're very right to begin with. You're very right. Malaria is only transmitted by the genus Anopheles. And if you talk to the school kids, like I did a few weeks ago, they were saying in unison, female Anopheles mosquito. So it has to be female, yeah. But yes, only Anopheles, the genus Anopheles. And which takes me back to what the question that was asked before about the modification and whether I support that. So who knows if we tamper and genetically modify Culex or Aedes, that we now start transmitting Anopheles. Now we're saying it's only Anopheles. I mean, we start transmitting malaria, but now it's only Anopheles. But after changes, tampering with them, you never know. These are the unknowns. And we cannot pretend that we know. We cannot pretend that we know. There has been a lot of talk about, even about COVID, SARS-2. There has been a lot of talk. I don't want to get into that, but I mean, it's in the public domain. So you never know what would happen when you now tamper with them genetically. They may become suitable to transmitting a disease they couldn't do before the changes. Okay. So what was the question about the Anopheles? Yes, it's only Anopheles.
Okay.
Yeah.
Another question from Lucy Mwangi. Mosquitoes are also pollinators. Are there specific species, perhaps the non-infectious disease-causing ones, that are pollinators? Is there a way to take this out, or has it been out already?
Well, even disease transmitting ones, not just non-disease transmitting, all mosquitoes, as long as it is visiting a flower for nectar, of course, it is doing the pollination. So I think her point was, her question, are there non-disease transmitting? It is not just the non-disease transmitting, even the disease transmitting that visit flowers, they are pollinators. I can't remember the last part of her question.
The last question was, is there a way to tease this out, or has it been teased out already?
About what? The pollination aspect? Yes.
Are there specific species, perhaps the non-infectious disease-causing ones, that are pollinators? I think you've answered. Yeah.
I guess I answered that.
Yeah. So we move down, and okay, there's a concern from Helida Oyeke, and asking if alcohol could be a mosquito repellent.
It's probably an attractant. It depends on where it is. If it's on the surface of the body, like sanitizer, probably yes. But if it's in the system, probably an attractant.
As well answered. I think actually it's alcohol that is in the system already.
In the system, maybe probably an attractant. Depends on where the alcohol is. If it's applied on the surface, like a sanitizer, I guess it will repel them. But only for some time, a few seconds, because it evaporates very quickly.
Okay.
And they'll be waiting a few meters for it to evaporate and then come back. Yeah.
Okay. And a question from Peter Kibobi. Hello, mosquitoes, just like snakes, are usually perceived negatively. What economic value can we get from these creatures, just like crickets? The other question is, why are mosquito bites painful?
Yeah, I think
are not. Yeah.
Yeah, I think the benefits I already mentioned, yeah, in the talk, and unless I guess Kenodia came a bit, joined us a bit late. Yeah, fine. They may not be edible like crickets. Yeah, people now know about the consumption of crickets and other invertebrates. So mosquitoes may not be that edible, but I have highlighted other benefits. Yeah, like the bioindication of a changing climate. Their food, we may not be eating them, but fish eat them. Yeah, you know, the dragonflies eat them. Yeah, and you know, dragonflies are also fed on by other things. So think of their role, their place in the food web and in the food chains, and how they maintain, you know, that functioning in ecosystems. There's nothing that is useless in this world. I keep on saying, including that cockroach that you see in your house and you hate so much. There's nothing that is useless. It must have, even if you don't eat them, as in consume them as food, they have a big role they play in the ecosystem.
Okay, thank you.
And some people actually also say that they help us regulate and control human population. Why it's not for malaria and other diseases caused by mosquitoes, the world be bursting with people. So they help us, you know, regulate the human population, as bad as it sounds, but they help us, you know, control the population of human. And also some of these nuisance creatures like mosquitoes, flies, if you like, they help conserve the environment in the sense that, or ecosystems, in the sense that there's some areas or habitats that humans don't venture into because of the nuisances. Yeah, especially the sesses and the mosquitoes. I usually give an example, the flies that we all had even in high school, the Miombo woodland in Tanzania. Yeah, people have not ventured there to cut all the trees to farm tomatoes and all this because of the flies. Closer home, we have the Lambwe Valley, I guess. Yeah, the same case. Yeah, people have not encouraged. And who benefits? The environment, the ecosystem, thanks to the nuisances from mosquitoes and other biting insects. Yeah, so view them that way. Yeah, see them that way. Even if they are not put on the table as cakes and ugali, yeah, they have so many other benefits.
Okay, thank you very much for that. From Vivienne, very informative talk. How successful is this vaccine against malaria?
I do not know because it's just come. Yeah, it is just come. So I think we will be knowing as time goes by. I think it's just newly introduced and is undergoing tests. Mm-hmm. Yeah, I hope it works. I personally hope it works. And since the question was how effective, yeah, I think it's safe to know for now. I do not know how efficacious it is. Yeah, probably I'll be learning same time as her, how efficacious it is. It's newly introduced.
Okay, great.
Yeah.
Another question from Mwesh. Do mosquitoes pollinate specific plants or they are generalists?
No, they are generalists. Yeah, they are generalists. Yeah.
Okay, nice. Okay, second last from John. I feel guilty now killing so many culicines unnecessarily. Taking out message at
Yes.
home. Okay,
Yes.
yeah, and last one from Rupi Mangat. Can Anopheles females be generically modified to be malaria free?
I think that is one of the attempts. Yeah, that is one of the attempts. And I know there was one research. This is not about genetic modification, but there's one very interesting research out of ICIPE where they were using another, microorganism that is so naturally found within Anopheles that was proven to block, the transmission of malaria. That is not genetically modifying, but such kind of things and such kind of research is ongoing to see whether you can, prevent the transmission of malaria by Anopheles, like she asked. Yeah. So the question is, yes, there's quite a lot going on, yeah, to modify this and this here and there, to here and here, to see whether you can stop the transmission by the Anopheles.
Okay, we have come to the end of sampling all the concerns, all the questions that were raised as the presentation was going on. And it is now 1 hour, 40 minutes, fantastic moment of touring the mosquito world that has really been very educative, very informative, and we have interacted enough and we have got to learn a lot. So thank you very much, Mr. Laban Njoroge, for that nice presentation. And also thank you very much, our members, our invited guests, for creating time to be with us this afternoon. I just have some 2 announcements to make. That on 25th of February, it will be a Friday, 2 weeks from now, we will be having another virtual talk about the Maasai giraffe. Actually, communication, you will receive communication from next week. And also to invite each and every one of you who will get time to join us for the Wednesday bird walks, which are actually combined with the waterfowl census that are taking place here in Nairobi. Actually, for this month of February, the program is there in the NatureNet newsletter that we released on Monday. And as well, next week on Wednesday, the venue will be in Nairobi National Park. We are meeting at the main gate at 8:30 a.m. Thank you very much, and I wish you all a nice weekend. Asanteni sana.
Thank you.
We now leave at our own pleasure.
Thank you for inviting me.
Thank you. You're most welcome.